Okay, let me try and
Okay, that looks like it's.
So do you want it to be you try?
I think me is like most important, but if you can hear me, that's
There's a summer baseball league last year. They have midnight games on Sunday. So it's like in Alaska actually, the summer's actually nice. It's all the same every day. Games start at like central off.
Even in England, it starts. It gets dark at night. So the smell, and I was at my brother's house, we had a lot of outside. He was sitting there chatting afterwards, and I said, I'm so tired. And then I looked at my watch, it was like 9:30, and it was still
That's good, dude. It's the official seating arrangement. Like, there's trouble if I sit here? Excellent. How do that? It's great. Yeah, looking for you. There were lots of signs. Yeah, lots. Yes. It's also true, but the first instance. Well, I just finished there. Slightly get Oxford, Cologne, Marseille, Palo Alto, Cape
All right, let's get started. Welcome all of you to Thesis Defense, and I want to particularly welcome his thesis committee, Mike Yaffe and Ian Cheeseman and Eric Barrowby, who had to come from afar and deal with traffic, and really all of Shui's colleagues and friends who are here. So I had to do lots of these. In fact, this is number one for two that I'm doing today. So I've had to think a lot about what to say. Shuyu is an MD PhD student at Harvard in the HSD program. There's been a number of students from that program that have been in the lab. And she has participated in an aspect of the lab which has been probably our most important work today, which is on understanding how nutrients are sensed by a major growth pathway, this mTOR pathway. And Shuyu broke open what was perhaps the biggest question that we had for actually a long period of time, actually since I started the lab. And that is, what are the actual proteins that directly sense nutrients, in this case, amino acids? And so we'll talk about the identification of what we think is actually the first sensor for this pathway, which I think was a milestone in the field. You'll hear more about that from her. One of the things that's hard when you have a lab, and I know many of you have and many of you will have a lab, is when you start to have people that are as smart and as hardworking as you, but actually think in a different way. And one of the biggest challenges you have is to not try to bend the will of that person to your own will. And I have to say, Shuyu was probably amongst the first students I had who fit all the laudatory characteristics that I said, but also thinks differently than I do. And I tend to be much more of a doer type, like just try it, see what happens kind of thing. Shuyu tends to be much of kind of a big brain thinker type of person who wants to sort of ponder and then do the perfect experiment. Now, I have to say we never agreed on this, because I tend to think that we know very little about biology, and so we actually can't sit and think about it. Well, I think she thinks that you could sort of somehow conjure up the beauty of biology. But I very much learned to respect this aspect of Shuyu and to appreciate it, and I think some of the people that she worked with did as well, even though I tend to have dislike for people that think like I do, which maybe is not so surprising. So for me, it's actually been a learning experience to have Shuyu in the lab and to be able to appreciate that aspect of someone and not want to throttle her all the time. Which sometimes actually is certainly a feeling that but I very much enjoyed working with her and seeing her take some of my advice once in a while, and me take some of her advice once in a while as well. And so she is going to talk about this project on amino acid sensing, but one of the things I admire about Shuyu is that when she completed this, she could have readily done probably a couple more papers based on the findings that she's going to present. But she decided that for her own training and for her own future, she wanted to do something completely different that we hadn't done at all, and maybe even something that she might want to follow up in her own future. And she's not going to talk about this project, but in fact she's done that really from the ground state, a project that she knew she probably would finish given the timings of going back to medical school. But I admire that she took sort of a long-term view to something rather than the short-term benefit that she could have. And lastly, Shuyu is a very interesting person other than outside of science. She's not one of these people that doesn't have other interests. And the one that I found the most fascinating is her passion for German football, which I had no idea about, particularly a team called Borussia Dortmund, which I happen to be going to Dortmund for, I have no idea why. I said I recommend to go, and I got her a cap for Borussia Dortmund, which was their team. And every time I see her wearing it, it makes me very happy to see that. So I have no idea why her allegiance is for that particular team. So I just Shuyu, look forward to your talk.
This is on. Okay. Thanks very much, David, for the kind introduction, and thank you, everyone, for coming to my Thesis Defense talk today. So today I'd like to share with you
some of the work we've done in the past few years on studying the mechanisms of amino acid sensing by the mTOR1 pathway. The discovery of mTOR is perhaps one of the most serendipitous examples showing how pharmacology can inform the discovery of novel biology. That pharmacological agent was originally isolated from bacteria in soil samples from Easter Island and was accordingly named rapamycin. Rapamycin was found to have profound inhibitory effects on immune cell proliferation and has since been used clinically to block the immune-mediated rejection of organ transplants, such as in kidney transplant patients here. The protein target of rapamycin, creatively named TOR, was finally identified in the early 1990s by, amongst others, David himself when he was a graduate student. And in the intervening years, mTOR has been found to be a major regulator of growth and metabolism across a wide range of species, ranging all the way from yeast to worms to flies and to mice. Now, given the importance of growth and growth regulation, deregulation of the mTOR pathway can have devastating consequences. So if deregulation leads to abnormally hypoactive mTOR, abnormally small body size could result, whereas abnormally hyperactive mTOR could give rise to cancers like glioblastoma, which is a disease of cellular overgrowth. It has also been increasingly linked to aging, to which a major contributor is thought to be a failure of metabolic homeostasis. And indeed, chronic administration of rapamycin is thought to prolong the lifespan in a number of model organisms, including the several that I mentioned on the previous slide. We are then interested in dissecting the molecular mechanisms behind the mTOR pathway in hopes of, one, understanding how it might go awry in disease, and two, identifying potentially druggable targets to modulate and to correct pathway behavior. mTOR, protein kinase, and its binding partners, which we collectively refer to as mTOR complex 1 or mTOR1, decides whether or not the cell ought to grow. And in this capacity, mTOR1 needs to be able to determine when conditions are permissive for growth, as well as to execute a growth program leading to biomass accumulation. Specifically, mTOR activity is regulated by local nutrient conditions, particularly the availability of amino acids, which serve, of course, as the building blocks of protein synthesis. And this will be the focus of much of my talk. So let's continue with the introduction. mTOR1 is also sensitive to energy, oxygen, and stress levels, as well as to circulating growth factors, which coordinate the growth of different cells in a multicellular organism. Now, when all of these upstream inputs are satisfied, mTOR complex 1, or mTOR1, can activate a suite of phosphorylation-dependent metabolic processes leading to the synthesis of proteins and lipids, nucleic acids, and energy, many of the key ingredients for growth in a cell. mTOR1 also inhibits processes that break down proteins and lipids, such as autophagy. There is a lot of work ongoing studying the mechanisms both upstream and downstream of mTOR. But for the purposes of this talk, I'm going to focus on the upstream pathways, namely the things that mTOR needs to be able to sense. So at the molecular level, mTOR1 activity is regulated by the actions of two small GTPases. The red GTPase, which is shown here in its inactive GDP-bound form, as well as the RAG heterodimer GTPase is also shown here in its inactive nucleotide configuration. Red is constitutively situated at the lysosomal membrane. And once activated, it can activate the catalytic activity of mTOR1. By contrast, the RAG GTPases, when active, promote the translocation of mTOR1 to the lysosomal membrane, where it can then be activated by red. So illustrated simply, when growth factors are present, energy is high and stress is low, red becomes GTP loaded and thus activated. But in the absence of amino acids, mTOR1 is nowhere near the lysosomal membrane to be activated by red. Once amino acid levels are sufficient, the RAG GTPases become loaded in such a way to recruit mTOR1 to the lysosomal membrane. And upon contact with red, mTOR1 itself gets activated, leading to a series of growth-related processes. And so in this process, the Reg GTPase and the Rag heterodimeric GTPases serve as two halves of a coincidence detector, ensuring that mTOR1 only becomes active when all of its upstream inputs are fulfilled. So now I want to zoom in on a particular part of this vast signaling network, which is how amino acids are sensed by mTOR1. Now, this question has fascinated scientists for many decades, but we actually didn't know very much about this pathway until recent years. And so in physiological parlance, amino acids actually refers to a series of 20 distinct small molecules. And the way they're sensed by mTOR may not be as straightforward and direct as the single arrow in the simplified diagram might suggest, but rather the sensing might occur through a series of signal transduction intermediates culminating at the Rag GTPases, which of course are the penultimate players responsible for recruiting mTOR1 to the lysosomal membrane. When we start thinking about how amino acids might be sensed by this major growth pathway in the cell, we start to realize this is actually quite a complicated question. For one, which amino acids are sensed? Is one amino acid sensed, or a subset of amino acids, or some generic property of amino acids involved? And if multiple amino acids matter, how are these multiple signals integrated? Secondly, at what concentrations are amino acids sensed? Unlike insulin, which is sensed in a relatively binary absent or present fashion, amino acid levels fluctuate but never to zero, since they're important building blocks in major cellular processes. So in this case, what are the concentrations that matter for sensing? 3, where are amino acids sensed? Are they sensed in the cytosol or cytoplasm, where protein synthesis occurs? Or might they also be sensed in specific membrane-bound subcellular compartments, or potentially as a gradient across membranes? And lastly, how are amino acids sensed? What are the proteins involved in this process? And are amino acids sensed in their abundance or in their scarcity? So before I proceed, I just wanted to briefly introduce a readout we use for mTOR1 activity. So as I mentioned before, mTOR1 is a protein kinase, and one of its main substrates is SXK. When mTOR is on, SXK is phosphorylated, as indicated by a band in this phospho-SXK blot. And so we simply use phosphorylation, or the degree of phosphorylation of SXK, as a readout for the degree of mTOR activity in general. Okay, so back to the first question I posed a few slides ago, which is probably the most straightforward of those questions. We found in an experiment that if we stimulate cells with a set of essential amino acids, they had the same activating effect on the mTOR pathway, shown here, as all amino acids. Conversely, if we added non-essential amino acids to cells, they had no activating effect whatsoever. This suggested to us that the amino acid signal to mTOR1 is fully contained within the set of 10 amino acids in the essential amino acid subset. We've also known for a long time that leucine and arginine, indicated by L and R, have particularly privileged roles in activating the mTOR1 pathway. But when the two are added in combination, they never fully activated the pathway, as indicated by this reduced phospho-SXK band in this lane. So in this experiment, we added a third essential amino acid to the LR pair, and noticed that the addition of lysine, or K, boosted mTOR activity to levels similar to that of all amino acids. Indeed, when we stimulate cells with LRK together, but not any two, we get this full activating effect on the mTOR1 pathway. This suggested to us that leucine, arginine, and lysine are sufficient for fully activating mTOR1. We also then did the converse experiment, in which we stimulated cells with cocktails of essential amino acids that lacked leucine, arginine, or lysine. And we noticed that missing any of these 3 amino acids was sufficient to fail to activate the mTOR signaling pathway. However, omission of any other essential amino acid, histidine, isoleucine, and a bunch of these amino acids had no effect on pathway behavior, or did not alter the effect of essential amino acids on pathway behavior. So So this indicated to us that LRK are also necessary for fully activating the mTOR1 pathway. Now, given these results, it would seem that mTOR is not simply sensing a generic property of bulk amino acids, since the molar content of LRK is much less than the molar content of all amino acids at RP1 levels. Additionally, the sensing mechanism appears to be relatively or highly discriminatory for side chain identity, as leucine cannot be substituted by isoleucine, valine, or methionine, which all have aliphatic side chains. Moreover, arginine and lysine, which are both cationic amino acids, cannot substitute for each other. And so this suggests that there might be multiple sensors, for example. And so why might the cell care about all the amino acids, LRK? So one possibility is, of course, there needs to be integration of multiple independent sensing events. Shown here. Perhaps 3 amino acids all need to be bound by their cognate sensors before the mTOR pathway is fully activated. Alternatively, it's possible that one amino acid might promote the entry of other amino acids into the compartment where sensing happens. We call this the gatekeeper hypothesis. So one formal possibility is that one of these 3 amino acids helps the others get across the plasma membrane into the cytoplasm. We have some metabolomics evidence to suggest that this is perhaps not the case. It appears that leucine, arginine, and lysine are not codependent for cell entry. When we add all amino acids, leucine, arginine, and lysine accumulate to these levels in the cell. And when we drop out any of these 3 amino acids, the accumulation of the remaining 2 is not dramatically altered. However, this does not rule out the possibility that LRK might still act as gatekeepers at subcellular membranes in specific compartments within the cell. And in order to test this hypothesis, we would need much more sensitive and robust methods, accurate methods, to determine intraluminal amino acid concentrations. So at this point, we were at a crossroads. How do we go forward, trying to find a sensor or potentially sensors for leucine, arginine, and lysine? We considered a few possibilities at this point. One, we thought about using potentially a chemical biology approach to use the sense amino acid to fish out potential sensors. We also considered taking advantage of our knowledge of the pathway, the protein components that have already been attributed to this process, as well as their subcellular localizations in the cell. And this indeed proved to be the way forward. So in addition to the RAG GTPases whose function in translocating mTOR1 to the lysosomal membrane I described earlier, our lab has also identified the regulator complex, or the regulator of the RAGs, which serve to anchor the RAG GTPases to the lysosomal membrane, and also to promote the nucleotide loading states that activates the RAGs. The lab has also found that the VTPase, which is a many-protein complex canonically thought to acidify the lysosome, is also necessary for transducing the amino acid signal to mTOR1. Now, previous work in the lab suggested that amino acids need to accumulate within the lysosomal lumen in order to activate mTOR1. And so, with this hypothesis, we decided to search for lysosomal transmembrane proteins that can simultaneously monitor the amino acid composition of the lysosome, as well as to signal to pathway components on the cytoplasmic phase of the lysosomal membrane. We identified a number of candidate proteins from our immunoprecipitation mass spectrometry experiments using RAGs and regulator as the bait. And the protein that most piqued our interest was SOC38. So what is known about SOC38? Well, one, it's ubiquitously expressed across all tissues. It's evolutionarily conserved to C. elegans or worms. It has homology to known amino acid transporters, yet its function and substrate specificity are not very well characterized. So topological predictions suggest that SOC38 might look something like this, in which it's with an N terminus, shaded here in purple, facing the cytoplasm, as well as 11 transmembrane domains across the lysosomal membrane. And this project has been in equal collaboration with a former grad student in the lab, Z. So first, we checked where SLC38-9 is localized in the cell. So when stably expressed in HEC293 T cells, SLC3889 co-localizes with LAMP2, which is a bona fide marker of lysosomal membranes, as shown here by this merge. This leads us to conclude that SLC3889 is indeed localized to the lysosomal membrane, consistent with the fact that it binds other proteins, RAGs and regulator, that we know to be at the lysosomal membrane. We also checked if SLC3889 interacts with known components of the amino acid sensing pathway, namely RAGs and regulator. Indeed, immunoprecipitation of full-length SLC3889 recovered amounts of regulator components P14 and P18 that are comparable to immunoprecipitation through the RAG heterodimer, which is our positive control. Curiously, pull down through the N-terminal domain alone, which is indicated here in this dotted box, was sufficient to recover regulator to the same degree as the full-length protein, suggesting to us that the N-terminal domain is what mediates the contact between this protein and RAGs and regulator. Since we're interested in mechanisms of amino acid sensing, we also wondered, might amino acids regulate the interaction between SLC3889 and these downstream components of the pathway? And indeed, this is what we find. In the presence of amino acids, the interaction between SLC3889 and the regulator components appear to be weakened, as indicated by reduced recovery of P14 and P18. Curiously, the N-terminal fragment of 3889 does not behave the same way, as in both amino acid starvation and stimulation conditions does it recover the same amount of regulator. This suggests to us that the transmembrane domain, which is present in the full-length protein but not in the N-terminal domain, could carry or could be responsible for the amino acid sensitive interaction. So far, as a summary, we think that there might be an amino acid sensitive domain in the transmembrane portion of this protein, as well as an N-terminal effector domain that plugs into the downstream players in the pathway. So next we wanted to test what are the functional signaling consequences for this interaction between 3889 and RAGs and regulator. When we overexpress a control protein, such as MTAP2, in the absence of amino acids, mTORC activity is off, as indicated by the absence of RAG here. And with amino acids, mTORC activity turns on, as we would expect. Interestingly, in cells that overexpress SLC3889, even when amino acids have been starved for or are absent, mTORC activity is still on. So somehow the overabundance of this protein tricks us into thinking that amino acids are there even when they're not. And interestingly, overexpression of the N-terminal effector domain has the same effect. In the absence of amino acids, mTORC activity is still on. This suggests to us that the interaction between this N-terminal domain and RAGs and regulator is probably necessary and sufficient for activating this pathway. But at the same time, this assay doesn't give us very much insight into the amino acid specificity of this interaction, since even in the absence of the transmembrane domain, because the N-terminal domain doesn't have that, which we think has the amino acid sensitive element, this N-terminal domain is already capable of fully activating this pathway. When we started to look at signaling in cells that have been depleted for SLC3889, we started to see some amino acid specific effects. In these cells, we knocked out SLC3889 using CRISPR-Cas9. And following starvation for arginine, which turns off mTORC activity, we noticed that adding back the missing amino acid to these different levels fails to activate the pathway to the same degree as in control cells or control clones. Interestingly, this is not seen for all amino acids, which we know to matter for the mTORC1 pathway. Shown here, leucine activation is not too noticeably altered even in the absence of 3889. So this suggests to us that SLC3889 might be interacting or sensing arginine more so than leucine. And at this point, we still haven't really shown that SLC3889 binds to amino acid, which we would think is a requirement for an amino acid sensor. So to address this, we decided to reconstitute SLC3889 in proteoliposomes and then test for its transport activity. In this experiment, SLC3889 in the context of proteoliposomes is exposed to radioactive versions of the amino acids listed here. We notice that SLC3889, curiously, seems to have a relatively broad substrate specificity. It has a preference for charged polar amino acids, but still is able to transport or at least allow these other amino acids to compete to some degree. So this leads us to wonder, how do we reconcile the apparent broad substrate or broad transport specificity of this protein with its arginine-specific sensing abilities? Well, one model is that perhaps SLC3889 is simply coming into contact with arginine more so than other amino acids at the lysosome. And there have been previous reports indicating that arginine concentrations are particularly high in the lysosome or in yeast vacuoles, which are evolutionarily related to the lysosome. And so perhaps SLC3889 is coming in contact with arginine and conveying its presence to it. Alternatively, it's also formally possible that SLC3889 has multiple binding sites for amino acids, one of which is specific for arginine, and another that's more permissive for a range of amino acids. And this model would then both account for its arginine sensing capabilities as well as its transport of a wide range of amino acids. Of course, we wonder, is there physical coupling of arginine binding and transport to downstream signaling through the N-terminal domain of SLC3889? To properly address this question, we need to have a very detailed understanding of the structure and structural conformation changes in this protein, but we don't have a structure of this protein at present. However, it's interesting to speculate what could be happening. So this is a schematic of a protein transporter distantly related to SLC3889. Amino acid binding is thought to occur, or at least the amino acid binding site is thought to be part of this transmembrane domain 1, which is most proximally linked to the N-terminal domain shown here by this purple dot. And one can imagine that binding of amino acids to transmembrane domain 1 and the subsequent conformational change that occurs with each transport cycle could alter the conformation of this N-terminal domain, allowing it to signal to downstream players in the process. So, in summary, we've shown that SLC3889 binds to RAGs and regulator, node components of the amino acid sensing pathway, in an amino acid sensitive fashion. We also show that overexpression of this protein's effector domain particularly, is able to activate the mTOR1 pathway, even in the absence of amino acids. By contrast, depletion of this protein affects arginine signaling more so than leucine signaling. And lastly, we've shown that SLC3889 can bind and transport amino acids. There remain, of course, many, many questions regarding the function and form of this protein. First, as I've already alluded to, is the transport activity of SLC3889 coupled to its signaling through the N-terminal effector domain, and if so, how does this happen? Well, to formally prove that this is the case, it would be good to identify transport dead mutants of this protein and see if this mutant fails to rescue the signaling defects of SLC3889 null cells. If so, then this potential model is a possibility. Secondly, is SLC3889 simply sensing the lysosomal contents, reading lysosomal contents? Or, being a transporter, does it also somehow alter lysosomal contents? And if so, how does this happen? We would like to know, of course, if the Km of SLC3889 for various amino acids, as well as the concentrations of these amino acids in the relevant compartments, jives with this model. Next, how does the effector domain of SLC3889, again shown here in purple, trigger mTOR1 pathway activation? To answer this question, we would probably need very carefully conducted biophysical studies, much like what Juan does in our lab, to figure out if SLC3889 is somehow directly affecting the RAG nucleotide state, or indirectly doing so by altering regulator function, or acting on RAGs after regulator has acted on RAGs. There are a number of possibilities here, but we would probably need an in vitro reconstituted system to dissect this process. And lastly, what is the physiological significance of SLC3889? So far, all of the experiments we've done have been conducted in cells in a culture or in a dish. But you can imagine that SLC3889 might have different and potentially interesting roles across different tissues of the body. And so, In order to address this question, it would be helpful to make models in which SLC3089 is knocked out or overexpressed or mutated in one or more tissues in a body. Okay, let's look at this first. So now let's take a step back after having identified the first putative sensor of amino acids for the entropy pathway. When I first started taking data lab in 2000, this is really all that we knew about the pathway. We knew that the RAGs were responsible for recruiting entropy to the lysosome, and that the regulators served as a scaffold localizing RAGs to the lysosomal memory. In the intervening 6 years, we've discovered a lot more about this pathway. So in addition to regulating the VTPs, which I alluded to earlier, through the work of Liron and Lynn and Rachel and Bobby, we now also know that there is this vast cytoplasmic arm to the pathway that culminates on RAG-A nucleotide loading state. Particularly, we now know that cestrins are cytosolic sensors for leucine, and that castor proteins are cytosolic sensors for arginine. And so, given the fact that we've now identified 3 sensors, or putative sensors, spanning 2 compartments, the cytoplasm and the lysosome, for 2 amino acids, leucine and arginine, we wonder why are there multiple sensors in multiple compartments for multiple amino acids? To begin with, why might sensing need to happen in both of these places? Well, presumably that's because amino acids are not in equilibrium between the cytoplasm and the lysosome, which then suggests that perhaps these separate amino acid pools have different physiological functions. It's perhaps a bit easier to understand why sensing in the cytoplasm needs to happen, since the cytoplasm is the site for protein synthesis, which is a highly energy-intensive process whose success depends on the levels of amino acids. Understanding why amino acid sensing might need to happen at the lysosome is a little trickier, although we could probably make a few hypotheses. First, the lysosome is, of course, the site of the release of newly liberated amino acids following autophagy or autophagic events. Moreover, it can serve as a storage site, a long-term storage site for amino acids. And this is indeed thought to be the case for yeast vacuoles, which are, of course, again, evolutionarily related to the lysosome. And so perhaps, given these 2 things, it would be beneficial to be able to detect contents of the lysosomal interior, especially if they're regulated differentially under nutrient starvation conditions. We also wonder, why are there so many amino acids? We've identified 3 of them so far, but we also know that there are probably independent amino acid inputs to the VATPase as well as to folliculin, which we think is a GTPase activating protein for RAG-C. And yet at the same time, there's still a whole bunch of essential amino acids for which we don't have a sensor. So if we subscribe to the notion that it's advantageous to be able to sense essential amino acids since they cannot be made in the cell, they're very physicochemically diverse and cannot undergo interconversion by definition, do these exist and where might they be? We already have good evidence to indicate that lysine is probably sensed in some way in HEC293 T cells, but we have no idea for these other essential amino acids in these cells. One possibility is that perhaps we need to starve cells of these amino acids for a much longer period of time, allowing the levels of these amino acids to drop significantly before we activate the sensor. There are, of course, also more questions. Given all of these sensors, are they all equally important, or is there a hierarchy? Might some sensors only become active once amino acid fluctuations are so extreme as to reach the KN of those sensors? Do different cell types or organisms express different sensors, and why might they care about different amino acids? So we do in fact know that this is probably true. Different cells and organisms care about different amino acids, but it's of course not yet clear why this is the case. And lastly, are these new pathway members or sensors deregulated in disease states? You can imagine for negative regulators of the mTOR pathway, loss of these genes might predispose to cancer. And there might also be more sort of wider range of metabolic derangements ranging from subtle to severe if there are mutations either in these sensors themselves or in the expressions of these sensors. For example, in our own experiments, we vary the does this happen physiologically or pathophysiologically? Okay, so that's about it for the data. I have many people to thank. This is just the start. I'd like to thank all of the people who were involved in studying SLC 3889, particularly my collaborator Z, former classmate, as well as Greg, who has taken over this project, as well as everyone who contributed with ideas and experiments during a particularly crucial time. I'd also like to acknowledge the people who have helped me work on a more recent project, which I haven't gotten to discuss today, particularly those who assisted me with setting up a function generating screen, as well as Nora, whose expertise has been invaluable in moving this project forward. There are also many current and former members of the Sabatini Lab I'd like to acknowledge. You guys have all contributed to my growth as a scientist, and it's been a pleasure interacting with you for the last 6 years in the lab. I'd like to thank members of my thesis committee, Professor Yaffe, Professor Cheeseman, and Professor Berkey for coming all the way from the US today. Many thanks for that. As well as the administrative support, the excellent administrative support I've received at many programs to date. Most recently, Betsy and Janice at MIT Biology, Patty and Rick at HSD, and Amy and Lauren from the Harvard MD PhD program. And I'd also like to acknowledge a few sources of funding, training grants, fellowships, and my research project. All right, now comes the fun part. This is a photo of our lab from fall of last year. We might look very serious here, standing in front of the white head, but I assure you that we also have fun, as you will soon see. In particular, I'd like to thank quite a few individuals. So first, Edie and Kathleen, our lab secretary and lab manager. Thank you for everything that you do. You make what we do possible. I'd also like to thank Roberto, who was my rotation mentor in 2010. He's now at Berkeley, so it's a lab there. Roberto is always very enthusiastic and positive, and I really appreciated those qualities, especially as he was showing me the ropes in my early days. I spent a lot of my time in graduate school in this room, 359. So sometimes we are working hard. I think there are about 9 transfer boxes between the 2 bays in these pictures. And sometimes you're hardly working, as you can see by these various sundry adventures, having dinner with Kika, baking with Liron, watching World Cup soccer, or snacking at Shake Shack. So I'd like to take a few moments to thank some individuals. First of all, Z, my MD PhD classmate from day 1, shown here dancing at the first year Society Olympics. There you go. Z, I've learned so much from you over the years, scientific and otherwise. And I particularly remember talking with you as I was trying to decide which lab to join during the second year of medical school. And I'm so thankful that we both decided to join David's lab. It's been a pleasure to have you as my baby and my deskmate, and I know that I've benefited from your unstoppable enthusiasm many points along the way. And these days, I'm really inspired by your drive to try to make science and medicine useful for humankind, and I wish you every success as you continue on this quest. I'd also like to thank you. I took over the SLC 39 project and has made tremendous progress in understanding how this molecule works. So you might be wondering what is going on in these photos. As it turns out, Greg is the runaway, lab-wide, perhaps even white-head-wide pull-up champion. And in this picture was taken just after he polished off 30 pull-ups, no big deal, and is standing there looking cool as a cucumber. So thank you, Greg, for being a wonderful deskmate for the past year, for regaling me with your encyclopedic knowledge of all kinds of music, and also for indulging me when I felt the need to nerd out about cycling, running, or music. I'd also like to thank Levin, who is one of the most unexpectedly hilarious and industrious people I know. So should you think that a stack of plates is tall or a collection of jewels that large is an anomaly, it must be an anomaly, I urge you to think again. Yeah, it's been a pleasure, getting to know you for the last several years, and even though we're both on our way out of the lab, I hope that we continue to find time for making adventures and walks around the trees. Rich and I are from the same town, which means that we're in Michigan, and we're also in the MD-PhD program together. It's been a joy working in the same room as you. Thank you for sharing your vast knowledge of podcasts of all kinds, as well as your heated political debates. I'm thankful we're usually on the same side. Rachel is also this picture I have of you doing your journal not too long ago. I'd also like to thank you for playing along with the Rich and Cookie game. This is one word that I'm sure you know well, and that has also greatly impressed your in-laws. So thank you for that at our new celebratory days. I'd like to thank who is the expert on all things quantitative in our lab. Fong is very industrious, although sometimes he's asleep at his desk. However, he's not. I particularly like this picture of three of my most recent bandmates, Fong, Ray, and Z, who were retreat in Savannah last summer. So thank you for putting up with all of our questions and for teaching me something new at every lab. I'd also like to thank two individuals, Tim and Kamanch, the screening gurus from our lab, for really helping me set up a functional genomics screen and showing me the pitfalls and ups and downs and what can go wrong and how things might go right. I'll briefly share an anecdote about Tim, even though he's not here today, because I think many of you might identify this in some way. So there are several things to know about Tim. One, he's very, very smart. Two, he really likes to talk. Three, he's fascinated by neuroscience. And four, he is a night owl. And sometimes these four things all converge, such that at 10 p.m., just as I'm about to leave lab, not wanting to spend yet another minute there, Tim walks into the bay and we start talking about neuroscience. And before I know it, it's midnight, and we're still in lab, and I have to get up for rowing practice in 5 hours, but we are still talking. And that's because the conversations with Tim always happen to be wide-ranging and incredibly interesting. So thank you, Tim, for these conversations, even though they came at the expense of hours of sleep. I'd also like to thank the LC members that Mike and Lisa set up and that Lisa now runs. Lisa, thank you for being willing to experiment with new protocols to detect amino acids in the SLC38 project, as well as lipids in this more recent project. I very much appreciate your rigor and adventurousness in this regard. Also, thanks to Tenzin for processing my samples with patience and care, especially when I submitted too many of them at once. I should say that this picture of Mike, despite the unusual headdress, is probably still one of the more recognizable photos of him. So thank you for this fast revenue knowledge. I'd also like to thank the dear friends I've gotten the chance to work with in room 329 over the past year, Walter, Nora, and Maria. Walter, thank you for being an awesome classmate for the past 7 years. I've learned so much from your probing questions and your curious mind, and I'm very, very excited to continue third year medical school at MGH on same 7 months of rotation as you. Thank you also for keeping us entertained during our many lunches, even if it cost some of us to starve our food from time to time. I'd also like to thank Nora. How wonderful it is that we're lab mates again. Nora and I were actually both in the lab in 2010. I was there as a rotation student, and Nora as a master's student. She recently rejoined the lab and has been working in room 329. Thank you so much for your expertise and collaboration on this more recent project. I've learned a lot from you. Thanks to Maria, one for recording today's talk so that my family can watch from home. And also thank you for many, many wonderful conversations. Really with all three of you. The talks are wide-ranging and perspective-enlarging, and I really enjoyed spending time in this back room. You guys have also helped me fulfill my years-long dream of pretending to be Freddie Mercury. So thank you for I'd also like to thank Joe as a dear lab mate until not too long ago, last fall. Thank you, Joe, for many, many incredible and eye-opening conversations about science and life over dinners at COSI. I'm so excited that you got to start your own lab, and I'm very excited to see how you progress science in the future. And last but not least, I'd like to thank David. So David is many things, amongst them silly, somewhat athletic, and also sure that we have fun from time to time in our lab, such as this past year when we started a series of alcohol tasting. David is kind of content or smug looking here, and the entire lab flocks around him, enjoying 4 different types of expensive whiskeys, including an 18-year gallon. But of course, David has also been an incredible mentor to me, to many of you over the years. Thank you, David, for teaching more than I could ever have imagined. Thank you for pushing me to try to be my best, and for trying to imbue me with a just-do-it mentality, even when I sometimes have the tendency to overthink and plod and all. So I really appreciate the rigor and carefulness that you've tried to instill in us, as well as the deep probing questions you ask us both in our one-on-one meetings as well as in lab meetings. I've learned a lot from you, and going forward, I hope to carry sort of rigor, clarity of thought, and this just-do-it mentality as I proceed my own scientific career. And I think many of you in the audience will also agree that some of our formative early scientific experiences have very important impacts on our lives. And I'm very thankful today to have a few mentors from the past who are here watching. So thank you to Dr. Brian Bowman, who was my postdoctoral mentor in Greg Verdine's lab for several years, for coming here today. I'd also like to thank an old mentor of mine from 13 years ago, Professor Mahanti. So Professor Mahanti knew me since I was essentially a kid, getting excited about science and science research, and it helped set me off on a path towards scientific discovery. I will always remember the exemplary model of mentorship that you embodied, and it means so much to me that we're here today. I'd also like to thank a bunch of dear friends, Priyanka, Benita, Matt, and Alex, who are my running buddies / coffee buddies / life coaches. We run marathon relays in the Harvard Stadium stairs. We've gone out for dessert and beer and pizza and dancing. We've also celebrated weddings and graduations. You guys have cheered me on when things got stressful, and you cheered me up when I fell down, and you've carried me through all kinds of ups and downs, and I'm so grateful for your friendship. I'm also thankful for the fact that you've occasionally let me steal the amp plate or 10 from the 6th floor. Next, I'd like to thank a good friend, Mike, or should I say Professor Mike Birnbaum, of the 3rd floor of this building. Mike is a former classmate and former neighbor and furniture moving assistant and grant application and thesis defense talk feedback giver. Mike apparently wears many hats. Thank you for many incredibly interesting, curious, and fascinating scientific discussions, even when they take a diabolical turn, and for generally being inspiring and encouraging. It's great that you're back in town. I'd also like to thank a bunch of truly wonderful and insightful, brilliant, and insatiably curious friends. I'll start with my econ friends. Professor Lagawa, Professor Scott Connors, and Gabriel Kreindler. You guys have really challenged and broadened the way I think, allowing me to see things in everyday life through new and interesting lenses. Particular shout out to Scott for being here today, I'm sure among the non-stop conferencing. Thank you for letting me sit in on your market design class last fall during a particularly wacky time in graduate school, and I look forward to continuing our collaboration with your registries. I'd also like to thank Rachna, Raj, and Hemi, the 3 members of my wonderful academic support group, for many wonderful monthly support group dinners and stimulating discussions, which have just been transformative, I have to say. Hopefully one day we will finally meet Mom. I'd also like to thank my friends from the MIT Rowing Club for 5 years of camaraderie and fond memories on and off the water. You guys have really taught me what it means to be part of the team. And I should say that in real life, no one has ever accused me of being too loud, or even loud, or aggressive, or bossy, or heart attack inducing, but apparently something changes when we set foot inside that boat. So really, thank you for putting up with me at 6 a.m. I'd also like to thank a bunch of classmates from various programs, my MD PhD classmates, my med school HST friends, and also, of course, most recently, my classmates in the biology PhD program. You guys have all been super supportive and inspiring, and I'm very much excited to see what you guys do in the future, and hope you continue to stay in touch. I'd also like to thank my dear friend Yao, who by this point has known me for more than half my life, for being a source of strength and wisdom over the years, particularly in recent months. And I'd like to thank my roommates, especially those, including Lucas, who was watching from the live stream in France. You guys have been really patient and supportive and fun, so thank you for that. And last but not least, I'd like to thank my family, shown here at my brother's high school graduation just 2 weeks ago. My parents are looking absolutely weary here, although surely relieved that nearly 30 years of child rearing is almost over. I'll start with my brother, Bryce. It's been incredible to watch you grow from an infant shown here on his first birthday to the quite amazing young person that you are today. Being your sister has by far made me a better person, and it's been one of the greatest privileges of my life. And just when MIT thought that I was finally getting rid of it after some 13 years, depending on how you count, turns out that my brother will be here 2 months as a college freshman. So good luck with him. I should also add that my brother chose this picture himself, which might tell you something about his priorities. And lastly, I'd like to thank my parents for what we call the mother and the baba. My parents, thank you for a lifetime of support and encouragement. Thank you for showing me through your own steady example that learning can be a lifelong passion. And thank you as well for the not-so-occasional and not-so-subtle reminders that fun and relaxation and rest are sometimes just as important as hard work. You guys have sacrificed enormously so that I can have the opportunities I've had, and going forward, I hope to continue to make you happy and proud. And that's it. Thank you.
Thank you, Shu. I had no idea you knew so many people. We have a couple of minutes for questions, but not in the committee, but so.
Yes.
Has the topology of SLC 39 been verified experimentally?
Yes. So what we can do is we can add epitope tags to either the N or C terminus, and we can also use. So then we can also introduce a cut site, for example, and see how I guess the experiments follow. So we have done these experiments, and it suggests that SLC39 is oriented in a way that the N terminus faces the cytoplasm. The C terminus ends somehow in the membrane of the lysosome. Yes.
How good of a transporter is in terms of flux compared to other studied transporters?
So that is a good question, and I think that question remains a work in progress. Initial experiments seem to suggest that this protein doesn't have very high affinity for amino acids, perhaps in the low millimolar range, something relatively high. So in this capacity, it's not entirely clear if it's a sort of high capacity transporter or if amino acids exist in levels high enough to get transported by this protein. The definitive experiment for that would be, of course, to introduce a mutation such that it can no longer bind, or getting rid of this protein and trying to determine if there are alterations in the amino acid contents in lysosomes or in the cytoplasm. So for now, I think I would say we're not entirely sure. We think that it doesn't bind to any amino acid with super high affinity, but its transport activity is still under investigation.
Thank you, everyone. And so, anyone wants to come by around 5 or so. If things go well here, we can do something on the 3rd floor. Thank you all.
How's everything going?